Hi, my name is David States. I'm a senior scientist at the National Center for Integrative Biomedical Informatics at the University of Michigan. I'd like to talk to you about some tools we've developed for analyzing molecular interactions. The Human Genome Project has revealed many thousands of genes, but the big challenge now is to understand how those genes function. And a key part of that is knowing what genes interact with each other in modules or pathways. To help analyze this, we've developed some software in collaboration with Trey Ideker at the University of California, San Diego. This is an application called Cytoscape that can be downloaded for free by anyone from cytoscape.org. Let me begin by installing a plugin we've developed to access our data at the NCIBI. So we go to the Plugins menu, and then we say Manage Plugins. And we have plugins available for download. We click on Network I/O, and then the MiMI plugin, Michigan Molecular Interactions, and just click Install. And we're going to the central repository, downloading the plugin, and now it's installed. And so now when we go back to the plugins menu, we see that our plugin's available. We click on Query, and we get a box that lets us query. I'll type in one gene name, CSF1R. This is a colony-stimulating factor 1 receptor that plays a central role in macrophage development. And let me make this a little bigger. These are the colony-stimulating factor 1 receptor and 19 genes that it interacts with. And we can try laying this network out in different ways by going to the Layout menu, for example, clicking Hierarchical. And we now get a network with CSF1 as the receptor as the central node, and different genes that it interacts with, including downstream kinases and phosphatases that regulate this. Let me go back to a circular layout here just so it's easier to see. We can also do things like take one gene, say the ligand for CSF1 receptor, and double click on that and get the genes that CSF1 interacts with, as well as CSF1 receptor, and keep going to expand this network as we are interested. We can also go and find additional information by right-clicking on this and linking out. So for example, we can go to our website about colony-stimulating factor 1 receptor, find additional information on the gene, annotations about its function, and a list with the different genes that it interacts with, and then look in more detail at that specific interaction, what sources we obtained it from, the actual publication describing the interaction, and that links out to the National Center for Biotechnology Information, NCIBI, and we see the abstract of the actual paper. And if you wanted to click here, we could go to the actual full paper itself. But this lets the biologist look at our data and dig down and say, where did we get it from? Do I really believe this data? And am I confident in using it in my research? We can also take one of the interactions, for example, the CSF1 with its ligand, link out there. And here, the NLP, we're getting actual sentences describing the interaction of CSF1 with CSF1 receptor. And again, the biologist can rapidly scan through this list and get a sense of in what context is this interaction occurring, what are the functions associated with it, and so forth. We can also make a larger network. I showed you initially just CSF1 and its immediate neighbors. We could go back and say instead of just the immediate neighbors, let's ask for a larger network of the neighbors and second neighbors. This will give us, again, it takes more time now because we're retrieving a lot more information. And what we'll get back is now a quite large network with several hundred nodes and several thousand interactions in it. In order to understand these complex networks, we need to decompose them and look at the structure within the network. And we see we retrieved now a network with 800 genes and 12,000 interactions. So it did take a little bit of time. And even making this as large as I can, it's obviously pretty difficult to understand. We can analyze this, however, by going to plugin called MCode. Analyze that. What we're doing is trying to find densely connected cliques or subgraphs within this network. And so MCode is now analyzing this large network. Its progress is indicated along this, this bar. And it will show us along this right-hand panel a list of subgraphs of densely connected gene sets that may have a coherent molecular function associated with them. All right, and now this is just completing. And we get a number of different subgraphs here. We can highlight one of those. If we say we wanted to see what was in that subgraph, we'll look at this data panel here, and we can ask for different features, a gene description, the gene name, what pathways and molecular processes it's involved in. And we can see this first subgraph that I highlighted is involving RAS homologs, is involved in MAP kinase signaling and a variety of biological processes. We click a second subgraph here. We now see we're seeing row-dependent signaling and continuing down, we can again find here a group of MAP kinase and MAP kinase signaling pathways. And it's a kind of paradigm that a gene tends to do what the molecules it interacts with. There are 20,000 genes in the human genome, and many of them have not been annotated. And so this inference about function through what an interacting partners do is a very useful paradigm. We can also blow this up and create a separate network for this. And so now we have a subnet that we can look at in more detail and see, in this case, a number of small G proteins in different ways of looking at that. But again, a fairly densely connected set of genes, which, when we look at their molecular functions, shows us that they do actually have a fairly coherent set of molecular functions in signal transduction. So in this way, the biologist can really explore the space of molecules and molecular interactions and learn more about the systems they're interested in and link out to other information resources. We're really enthusiastic to have people using these tools and giving us feedback on how we can improve them and extend them. Thank you, and enjoy looking at the tool.